All right, let's look at our last form of passive mechanism, and that is osmosis. Osmosis is the movement of water from areas of, Osmosis is the movement of water across semipermeable membrane from areas of low concentration of solids to areas of high concentrations of solids. Okay, so what is a solid then, right? So a solid is part of a solution. And a solution is a mixture of two or more substances. So one part is a solid, that is a dissolved substance, and the other part is the solvent. That is the substance that does the dissolving. So just an example here, right? So of a solution. So we've all probably made Kool-Aid at some point in our life. And make Kool-Aid, you get a pitcher, you put some water in there, you put some Kool-Aid mix in there, you put some sugar, and you stir it up. So here, the Kool-Aid mix and the sugar are being dissolved in the water. So here, the Kool-Aid mix and the sugar are solutes, and the water is the solvent. And when we talk about living organisms, water is the solvent of life. So things are dissolved in water in the cells. All right, so let's go back to the definition here. So it's a movement of substances across a semipermeable membrane, like our plasma membrane, from areas of low concentrations of solids or dissolved substances to areas of high concentrations of solids or dissolved substances. So water is going to move from where there's few dissolved substances to where there's more dissolved substances. All right, so this is how it works. So here we have some water, we have some dissolved substances in there. And so if you take this, you put it into the water, water will move into that tube to try to equal out concentrations there. All right, so here, this will occur until concentrations equal out. All right, so something I said about diffusion as well, right? The difference here is to equal out those concentrations in osmosis, here the solvent moves, in diffusion, the solid moves to equal out those concentrations. Now, so we're talking about water in relationship to solid concentrations. So this gives us, and we're talking about water is going to shift in those situations. So this brings us to three different types of solutions. Solutions that are the same concentration, solid concentration as our cells, some solutions with more and some with less. So the first is called an isotonic solution. In an isotonic solution, so iso, if you remember from your geometry class, an isosceles triangle has two equal sides. Iso means same. So, and tonic refers to solution strength. So an isotonic solution is a solution with the same or equal concentration of solids as body cells or body fluids. This is equivalent to a 0.9% NaCl or salt solution. All right? So if you took a cell, a red blood cell, and you place it into an isotonic solution, there would be no net movement of water, meaning water would enter the cell, water would leave the cell, but the same amount of water that entered the cell would be the same amount that left the cell. Okay? So, oh, by the way, for those of you who wear contacts, your saline solution is an isotonic solution. It'll say so on the packaging, or it'll say it's a 0.9% salt solution. Next is a hypertonic solution. Hyper, if you're hyperactive, your activity level is above normal. So a hyper is above. So this is a solution with a higher concentration of solids than cells. So this is above that 0.9% salt solution. So if we took a red blood cell and we put it into a hypertonic solution, because there are more dissolved substances outside the cell than within the cell, water will leave the cell to equal out those concentrations. Okay, so here that cell would shrink. All right. Oh, and this is what ocean water is. Ocean water is a hypertonic solution. Oh, by the way, yeah. Things taste good for a reason. Either they have nutrients that we really want in them, or they're high energy of some sort, right? So, you know, like ice cream tastes really good because it has sugars, it has fats, and we love both of those things. And some people really like it if they put a little salt in there, that salty ice cream. All right, but if you get too much of something, well, you don't like the taste of that though. And that's what we get with the ocean. It's a hypertonic solution. So it's anywhere from 2.5% to 3% salt solution. So if you ever heard you can get dehydrated from drinking the ocean water, that's exactly what can happen. So we drink it regular tap water, we absorb that water into our body. But if we drink ocean water, because of the hypertonic solution, we actually lose water from our body to our digestive tract. And then what happens with that is we poop it out. So if you continue to drink ocean water, what you will happen here is you'll die of dehydration due to diarrhea. So not a pleasant way to go. Okay, so the last type of solution is a hypotonic solution. So if hyper is above, hypo is below. So this is a solution with a lower concentration of solids than cells. So less than a 0.9% salt solution. So if we take a red blood cell and put it into a hypotonic solution, because there are more dissolved solids within the cell than outside the cell, water will enter the cell to equal out those concentrations. So, no idea why I just did that. Okay, so we go to this picture. It shows these three different solutions and arrows indicating the movement of the water. Isotonic, arrows in both directions. Hypertonic, water leaving the cell. Hypotonic, water entering the cell. And you can see that red blood cell will burst in a hypotonic solution. That is true, right? But the thing is, the only time a red blood cell gets into a hypotonic solution is if you take red blood cells out of somebody and you put them into a hypotonic solution, right? So that's not the normal situation. This is not going to happen to all of our cells. The cells that line our mouths and our digestive tract are not going to burst when put into a hypotonic solution. And how do I know this? Because anytime I drink water or tea or milk or anything like that, that's a hypotonic solution and my cells aren't bursting. In fact, all freshwater environments around us are hypotonic. So lakes, ponds, streams, stuff like that. All right, so let's move on to active mechanisms. So here we're going to use energy to get substances either into or out of cells. So the first is active transport. So active transport is a process that moves substances across a membrane from areas of low concentrations to areas of high concentrations. And this is going to use a carrier protein. So we go back to that definition. Move substances from low concentrations to high concentrations. So as we say in the definition, we can see that it's moving against diffusion. And because it's moving against diffusion, that's why it requires energy. So here, we pump stuff out of cells, pump stuff into cells, and it's going to require some energy to do that. Okay? So, one of these that we're going to talk about later on this semester is a sodium-potassium pump. Very, very important. If this thing stops working, we're going to stop working really quickly. Okay. Next is membrane-assisted transport. So here we're going to use a membrane to get stuff either into or out of cells. And the first is exocytosis, and this is showing exocytosis. So exo, exiting, cyto, the cell. So this is a transport of substances out of a cell by a vesicle. So here we have something in a vesicle. What's going to happen here, it's going to fuse with the plasma membrane. The plasma membrane is going to open up. It's going to release those contents out of the cell. This is how we get saliva out of salivary glands, tears out of lacrimal glands, and sweat out of sweat glands. If we're going to go in the opposite direction, this is called endocytosis. Endocytosis is transport of substances into a cell by a vesicle. So the first type here we're going to look at is called phagocytosis. Phago means to eat. So this is endocytosis around a large molecule or another cell. So here, right, the cell is engulfing this smaller cell. So this could be a white blood cell engulfing a bacterium here. And then a lysosome would fuse with that, and that would kill that bacterium. Next is penocytosis. So if we look at penocytosis, penocytosis is endocytosis around a liquid. So here's penocytosis, and this is showing moving this fluid to the other side, and you get exocytosis there. You can see this occurs along capillaries. It also occurs along our small intestines there. Oh, peno means to drink. So penocytosis, moving a liquid. Lastly is receptor-mediated endocytosis. So in receptor-mediated endocytosis is endocytosis around a specific molecule. So this is going to use receptor proteins. So that molecule binds to receptor proteins. When you get enough of them, we're going to pinch those off into a vesicle, draw them into the cell. This is how we get cholesterol out of our blood. So two ways, by our liver cells. Our liver cells pull that cholesterol out of our blood. Two ways to have high cholesterol levels. One is a bad diet. The other is poor genes. If you don't make a lot of these receptor proteins, you're going to have genetically high cholesterol levels.